Hello, my dear students. Welcome to a new episode of biology. Today we will talk about meiosis. The key concept here is during meiosis, diploid cells undergo two cell divisions that result in haploid cells. And we already know that the term diploid means the cells have the complete number of chromosomes, but haploid means the cells have half the number of chromosomes. We have two main ideas. Number one, cells go through two rounds of division in meiosis, unlike mitosis. Number two, haploid cells develop into mature gametes. Let's see. Cells go through two rounds of division in meiosis. Meiosis is a form of nuclear division that creates four haploid cells from one diploid cell. So the end result of meiosis is four haploid cells from one diploid cell. This process involves two rounds of cell division, meiosis 1 and meiosis 2. Each round of cell division has four phases, which are similar to those of mitosis. Do you remember prophase, metaphase, anaphase, and telophase? These four mitosis, they are similar in meiosis. Okay, to keep the two processes distinct in your mind, just to focus on the big picture, what happens as an overall view. Pay attention to the way meiosis reduces the chromosome number and creates genetic diversity. So let's start. The homologous chromosomes, which are shown as duplicated, they are two separate chromosomes, one from the mother and one from the father. This is why they are in different colors, just to indicate that they are not from the same parent. Okay, homologous chromosomes are very similar to each other since they have the same length and they carry the same genes, but they are not copies of each other. In contrast, each half of a duplicated chromosome is called a chromatid. Do you remember the sister chromatids? Together, the two chromatids, they are called sister chromatids. As you can see, these are two sister chromatids from the mother and two sister chromatids from the father, as example. Thus, the sister chromatids refer to the duplicated chromosomes that remain attached by the centromere, the central part. The homologous chromosomes are divided in meiosis 1. Homologous chromosomes, as you can see, they are divided in meiosis 1. And the sister chromatids are not divided until meiosis 2. Look here, the sister chromatids, they will be separated in meiosis 2. So let's talk about these processes in detail. Meiosis 1. Before meiosis begins, the DNA has already been copied. Yes, this is before any division. Meiosis 1 divides homologous chromosomes, producing two haploid cells with duplicated chromosomes. Like mitosis, scientists describe meiosis in terms of phases. The figure is simplified by showing only four chromosomes. Let's see. Meiosis divides the homologous chromosomes. So we have prophase, metaphase, anaphase, telophase. This is for mitosis, but here, as we divided meiosis into meiosis 1 and meiosis 2, so each phase has its own number. Then in prophase 1, now we are talking in meiosis 1, this is prophase 1. What happens in prophase 1? As you can see, the nuclear membrane breaks down, the centrioles and centrosomes begin to move to the opposite sides of the cell, and the spindle fibers start to assemble. Then the duplicated chromosomes condense and the homologous chromosomes begin to pair up. As you can see here, two chromosomes from the mother and two from the father, they begin to pair. Then the second phase is metaphase 1. In metaphase 1, the spindle fibers align the chromosomes, the homologous chromosomes, along the cell's equator in the middle part of the cell. Each side of the equator has chromosomes from both parents. So this is not alignment for the chromosomes from the father in one side and the chromosomes of the mother in the other side. No, they are mixed in between. Then what happens in anaphase 1? The paired homologous chromosomes, they are separated from each other by the spindle fibers and they move to the opposite sides of the cell. The sister chromatids remain attached. As you can see, this is a complete chromosome. Two sister chromatids attached to each other. Then in telophase 4, the spindle fibers break apart or disassemble. Then the cell undergoes cytokinesis. Let's talk about these processes in detail, these phases. In prophase 1, In early meiosis, the nuclear membrane breaks down. The centrosomes and centrioles move to the opposite sides of the cell, and the spindle fibers start to assemble. Then the duplicated chromosomes condense, and the homologous chromosomes pair up. Listen carefully. They appear to pair up precisely, gene for gene, down their entire length. The sex chromosomes also pair with each other, and some regions of the DNA appear to line up as well. Then anaphase 1. The homologous chromosome pairs are randomly lined up along the middle of the cell by the spindle fibers. The result is that 23 chromosomes, some from the father and some from the mother, are lined up along each side of the cell equator, as we said. This arrangement mixes up the chromosomal combinations and helps create and maintain the genetic diversity. This is for the sexually reproducing organisms. Since the human cells have 23 pairs of chromosomes, meiosis may result in 2 to the power 23 or more than 8 million possible combinations of chromosomes. Can you imagine the genetic diversity? Then anaphase 1. The paired homologous chromosomes separate from each other and move toward the opposite sides of the cell. The sister chromatids are still together. They remain together during this step, anaphase 1, throughout meiosis 1. Then telophase 1. The nuclear membrane forms again in some species, and the spindle fibers disassemble, and the cell undergoes cytokinesis. The end result of meiosis 1 is two cells that each have a unique combination of duplicated chromosomes coming from both the parents. Then let's move to meiosis 2. Meiosis 2 divides the sister chromatids that remained attached in meiosis 1 and results in undoubled chromosomes. The following description of this process applies to both the cells that are produced from meiosis 1. Note that the DNA is not duplicated again between these two stages. Let's see, prophase 2. Prophase 2, the nuclear membrane breaks down again, the centrosomes and centrioles move to the opposite side of the cell, and the spindle fiber assemble again. In metaphase 2, the spindle fibers align the 23 chromosomes at the cell equator. Each chromosome has two sister chromatids at this stage. Then anaphase 2, the sister chromatids are pulled apart by the spindle fibers from each other and move to the opposite sides of the cell. In telophase 2, finally, the nuclear membranes form around each set of chromosomes at opposite ends of the cell. The spindle fibers break apart and the cell undergoes cytokinesis. The end result of meiosis is four haploid cells with a combination of chromosomes from both the mother and the father. Look at this picture again. This is prophase 2, where centrosomes and the centrioles move to the opposite side of the cell. The nuclear membrane started to break apart again, and the spindle fibers started to assemble. Then metaphase 2, now we have chromosomes. The spindle fibers align the chromosomes along the cell's equator. These are two sister chromatids still attached by the centromere. Then in anaphase 2, the sister chromatids, they are pulled apart from each other and move to the opposite sides of the cell. Then telophase 2, the nuclear membrane started to reform again around the chromosomes. The spindle fibers break apart and the cell undergoes cytokinesis. And here are some differences between meiosis and mitosis. You remember in mitosis, the cell has only one cell division, but in meiosis cells, the cell has two cell divisions. In mitosis, homologous chromosomes never pair up, they align at the cell equator. But in meiosis, homologous chromosomes pair up along the cell equator. This is in meiosis 1. In mitosis, in anaphase, the sister chromatids separate, yes, but in meiosis, in anaphase 1, the sister chromatids remain together. In mitosis, it results in diploid cells. Yes, they are copies of the original cell or the parent cell, but in meiosis, it results in haploid cells. Then the haploid cells develop into mature gametes. Haploid cells are the end result of meiosis. Yet these cells are incapable of fertilization until they go through more changes to form mature gametes in the reproductive organs of any organism. A process named gametogenesis is the production of gametes. Gametogenesis is the production of gametes. Gametogenesis includes both meiosis and other changes that produce a mature cell. The final stages of gametogenesis differ between the sexes. The sperm, for example, the sperm cell or the male gamete is much smaller than the egg, which is the female gamete. The sperm cell's main contribution to an embryo is the DNA. As you can see, this is a germ cell that divide, undergo through meiosis. This is the germ cell is a diploid, undergo through meiosis. It produces four mature haploid gametes, which are the four sperm cells. Yet the sperm cell must swim to an egg to fertilize it. So the ability to move is critical. This is very important for the sperm to move. Sperm formation starts with a round cell and ends by making a streamlined cell that can move rapidly. This is streamlined shape, so this to help in making the sperm cell move rapidly. During this process, significant changes occur. Like what? The DNA is slightly packed and much of the cytoplasm is lost, resulting in a compact head. As you can see, this is the structure of the sperm. A head, then the middle piece, or this is the neck, and the flagellum or tail. The sperm cell develops a whip-like flagellum and the connecting neck region packed with mitochondria that drive the cell. The mitochondria here is very important because this is like the power plant of the cell. This is what gives the sperm the energy needed to move. Other changes, such as the addition of new proteins to the cell membrane, also take place. Then let's talk about the egg. The formation of an egg is a complicated process. As you can see, this is also a germ cell diploid in the female, undergoes meiosis. But here, the division of the cytoplasm is not even. They are not equal to each other. Then it results in mature gamete, haploid, one egg, and polar bodies. Let's talk about this. It begins the formation of an egg. It begins before birth inside the developing body of a female embryo. And it is not finished until the egg is fertilized by a sperm many years later. The process includes periods of active development and long periods of inactivity. An egg not only gives its share of DNA to the embryo from the mother, but also it contributes to the organelles and the molecular building blocks and other materials an embryo needs to begin life. This is very important. Only one of the four cells produced by each round of meiosis actually makes an egg. One cell, which is the egg, receives most of the organelles, cytoplasm, and the nutrients. This is why we said that the division is not even in case of formation of eggs. Many molecules are not evenly distributed throughout the egg's cytoplasm. This unequal distribution of molecules helps cells in the developing embryo to specialize. What about the polar bodies, the other cells that are produced in the process of producing eggs? The other cells produced by meiosis become polar bodies, which are cells with little more than DNA that are eventually broken down. In many species, including humans, the polar bodies produced by meiosis 1 does not undergo meiosis 2. And here we come to the end of our session. Hope you understood this part. See you in the next episode. Thank you guys and goodbye.